And to conclude our session this afternoon, we have another interesting talk from Federico Marini, representing Bioconductor, which is awesome. We haven't seen too much from Bioconductor yet, but there's really a wealth of very useful packages available in Bioconductor, which have applications even outside of the genomic and biomedical spaces. In this case, though, we are going to have a genomic application. Federico is going to show us some shiny applications that he's developed to allow us to explore genomic data, and in particular, RNA sequences. So it looks like Federico's ready, and I'll turn it over to him. Thank you, Federico.
Hi, thank you. I can't find the words to say how happy I am to be here. I have familiar faces, new faces. But I have a problem. I realize that I'm the obstacle between you and finding out the composition of the drinks on the cruise ship afterwards. So I will try to make it compelling for you. And don't forget to acquire some replicates of these measurements later. So what I want to talk to you about today is, first of all, a few words about me. I am a virtual postdoctoral fellowship at the CTH at the University Medical Center in Mainz, and I got my PhD in bioinformatics and biostatistics at the IMBY. The topic was to develop application for interactive and reproducible data analysis, and these were a combination of methods and tools that I wanted to construct, to design, to maximize information extraction in such a way that information translational interactions are made easy, and knowledge transfer is also coming along. I posted my slides over here, so I don't know if you can read it. Otherwise, you can also check it on from my Twitter handle. Right now, I think there's a lot of data available, way too much probably, so that we are actually not able to understand it properly if you lack the proper analytic skills. So in the ideal world, you have a wish list for accessible and robust data analysis, and you want it to be comprehensive, of course, so workflows in Bioconductor are very nice as a tool. Interactivity, because it empowers the domain expert and allows them to get better insights on their data, and technical reproducibility, so the ability to reperform the same analysis with the same code and getting the same exact thing. It's the essential component for having something transparent that can allow you an independent verification of the data and to build on the shoulder of other giants that have, say, built some nice methods and you want to develop further them. And this is particularly true in the field of bioinformatics. So in RNA sequencing, I don't know how many of you are familiar with that or have heard about it. So it's not a Bioconductor conference, but I can still think that some of you might be, at least have heard about transcriptomics and stuff like that. Few hands, but still some hands. So try to imagine that you have a sample, and in each of these samples you acquire a snapshot of 20 or 30 or more thousand genes. So if this is just one case, and it gets complicated because you have more and more and more of these genes, and you have more of these samples. So back then, a few years ago, you just have a couple of samples, and it was not so optimal. Now you tend to have a lot of data, as I said, and the aim, for example, one of the most common workflows is to identify the differential expressed genes, so which means the signatures, so the underlying reasons why, for example, you observe different phenotypes in your samples. I have been working also in my PhD thesis on platelet transcriptomics, which is per se already quite a cool thing. I don't know how many of you did know that platelets don't have any nuclei and yet have a ton of transcript inside of them. And it's not just the tiny little friends that help you when you get wounded and close up your wound nicely, but they also might be used as a liquid biopsy marker, for example, for cancer and metastasis. That's currently one of the applications for those. I've been doing a lot of exploratory data analysis, a lot of differential expression analysis, and I wanted to analyze, visualize, and integrate that, and I wanted to find a way to do it in a proper and compelling way. So, as I said, I've been working with quite some scenarios, which were, for example, from the lab of Professor Wolfram Ruf, which is now mentoring me also for the postdoctoral fellowship. And these will not be at all possible if the Bioconductor project wasn't there. So I've been a user for a while, and then I decided that it was my time to contribute something. So I provided this PCA Explorer package for interactive visualization using a principal component approach, and then IDEAL came along. So if you were wondering why I was using that word in the title, that's the reason. I wanted to do that. And basically, they are web-based applications that enable interactivity and reproducibility at the same time. What's going on under the hood, so which is most important for you? This might be your data, for example, and it's going to be just a table, basically, with some information on the metadata. Under the hood, it's R bioconductor, Shine is a framework, and then I'm exploiting the power of R Markdown to generate beautiful reports. So basically what I'm doing is I'm encompassing the whole workflow, and I'm encapsulating a template report in the Shiny app. And this basically picks the live inputs from the user interactions and generates these nice and beautiful reports. Why did I start reinventing this wheel? Because indeed, data exploration was somehow underweighted, and yet it's a very much essential step, especially when you do it in an interactive way. And something that indeed was fully integrated in Bioconductor. There were Shiny apps for doing PCA, but it was just using the iris dataset, for example. So I wanted also some transparency and help for my future self. And of course, I wanted automatic reporting and state saving. But that was before, say, Shiny nicely added the bookmark features. These would have the aim to empower the domain expert, and it's indeed used by some of cooperation partners, and they just don't want a list of genes, they just want more. And so we try to cover all the way from exploration to functional interpretation. One thing that was also nicely touched by Mikolai a few hours ago, is that also it's not just a platform for reproducible analysis, but you want some way to actually tell the user how to use your app, and it's not just text. So I also have interactive tools for some kind of self-paced learning. You try to force the user to do things in a correct way so that they actually know that they're doing the right thing. So in a glance, it just looks like this. You just have a couple of samples, and then you just launch the app. So cheating. It's not a demo in this case. I will just play out some demonstration. I think that probably it's not that useful now because it's quite small, but I had 13 samples with 2 different conditions, and I wanted to check, for example, how it looks like. For example, just a quick overview with the normalized counts. You just provide the row matrix of counts, and then everything is computed on the fly. You can do, for example, a distance between the samples so that you can find out, for example, you had one sample that looked very different from the others, and I wanted to check that further because sometimes it happens that when you sequence platelets, then you might have some contaminations, and indeed, integrin alpha M was just here, and that was already a clue that we probably needed to remove that sample for further analysis. We did that, so we checked, for example, also the principal component view of that, and we see that indeed it stands just right there, lonely, and we have good reasons and say some kind of support for removing that sample out of the analysis. So indeed, we can check with the outlier identification tool what happens if you just remove that, and indeed the situation is just getting better. Not perfect, of course, but because it's quite a noisy data set, but indeed it was a good starting point. So it's also robust to the number of genes that you include for the computation, and then we moved on to the differential expression analysis. So this is a screenshot of how ideal looks like, and it's structured in a slightly better way so that you can see, for example, you have sequential steps. You go from red to green through yellow, so that while you follow the workflow of the eSeq package, which is very widely used, you also insert some annotation objects, and when you run it, so all the things that you need are just shown in a kind of a better way with the Shiny dashboard. You can compute the results, you can extract them, and then once you do that, you can, for example, already obtain some kind of diagnostic plots on those, and nicely HTML-based. So this is just a quick peek in how the tools might look like. I will touch more upon that later, but it's just inviting the user to do the things so that he actually does that, or he or she does it, and then it just gets the things that he's supposed to get. I'll just skip a little to the part where you actually get, for example, some plotting, so you can obtain some overview in the form of heat map or interactive heat map. And if you just click on one plot, you can link it to the others and get some information. You might have your signature of interest that you want to see, for example, thrombospondin, you get it here, and then you just get, with auto-completion, the name of the genes over there. Whatever you did live is captured then by a template report, which is provided alongside with the package. And that kind of re-renders every time. I just skipped to that part. It renders live in the app, so you can, for example, have a preview over here, and then once you're done, you can just save it and share it and keep it, and or just build upon that. So back to the main one. We've been talking about bulk RNA sequencing. What you do is that you get a bunch of cells over here or over there, any kind of sample. And now technology in genomics has been so advanced that you can actually take single cells and sequence those. So instead of just having samples, so genes times samples, you end up having genes times cells. And data exploration has probably never been so important as of now, because a lot of hidden pearls are just inside your data already. You just need to take them out. So the problem is that all of this identification of subpopulation or describing the developmental trajectories, it just takes time, a lot. And you might need many iterations to do that. And then you might be in the difficult situation where you are at the end of your exploration, and then you ask yourself, how did I do this plot? How can I send this to my supervisor or stuff like that? So in December 2017, the whole thing started. So the whole inception of the project was December, yeah. And so it's a guy from Hong Kong, which was academically raised in Australia, a Swedish girl in Switzerland, a French guy in UK, and an Italian guy in Germany. It sounds like the beginning of a joke, but it's just say the description, probably the best accurate description of our team that came along. And we sat together, we made up a wish list of what we wanted to do. We wanted interactivity. We have it with Shiny. We wanted to link information across plots, which is very important, for example, in this. We wanted reproducibility, and we wanted to make a hammer for many, many nails. And a few months, at about a couple, thousand of commits later, we're still in the process of finding a proper hex sticker, but this is the draft version of it. If someone of you has better experience in designing those, I'm happy to take some suggestions. But if you want to see the bleeding edge, the very much bleeding edge, you can see it over here. So the link, again, is in the presentation that you can get afterwards. And if you want, you can be ready for a demo. So do I have some time? OK, that's great. So I'm not expecting you to reproduce your complete analysis. You should do this, but I'm not expecting you to just know that. I can show you that it's just easy to get it live. And to get it live, you can get it like this. So the app starts and takes basically just a summarized experiment as an input. And you can do many plots. You can have reduced dimension plots, column data plot, row data plot, feature assay plot, so whatever expression values. And you can pre-initialize it. So that's what I did, for example, with these 300 lines of code. Thing is, how do I get these lines of code? The answer is quite easy, and you can extract the R code with the magic wand over here. Whatever you did, it's just saved. So all of these things that you see here live, that I will show you just in a second more, you can just copy-paste that into your script, and you get the exact same things. Also, with the interaction that you had. So you can interact, for example, with your cells over here, and you can see that the selection is just changed in the other one, so I'm just selecting a now not so meaningful example. And if the RStudio guys are listening now, if they could implement this very nice lasso freehand selection tool, could somehow solve the problem behind this quite clunky but still working solution that we got for, okay, not working now, of course, but you can, trust me, when the polygon is closed, then the selection is just highlighted, okay. It can get complicated because you can get many charts, but you can also have the fact that all of these charts are linked together, and you see that from this graph. Yes, so you can open up, link all the plots together by these brushing actions. So you are getting that hammer for many nails that I was talking about. As an outlook, I mean, you've seen that there's a lot of flexibility and customizability for doing this. And the idea that we have, or we anticipate, is that you don't need to reinvent the wheel for a cool data explorer for the data that you have published in a journal. You can, for example, set up an instance of this, and then you have a live browser where you can reproduce, for example, the findings of your manuscript, and on top of it, you allow the user to explore your data fully. So if it's the case, for example, when you try to do that thing for the first time, you can somehow embrace the view of is it the most interesting man in the world, this meme? I don't know the exact name of it. So, as a summary, the main aim of this whole project is to make analysis and exploration accessible to many scientists, especially the domain experts, so it makes it accessible and still upholding the standards in doing reproducible research. It can become, these applications can become your best friends when you do RNA-seq and or other omics analysis. For example, you can also do mass cytometry with these. We all love bug reports, and even more. If you want to provide a pull request with some new features that you want to be implemented, we are happily welcoming them. And for me, for my outlook, for my long-term outlook, what I want to do is to have these apps as cornerstones for building up a portal which will include the many data sets that are available for RNA-seq in platelets. And on top of it, say, as a side effect, nicely, it's something to build something that actually try to get people from different worlds coming together and then somehow build a common ground for promoting the openness of science by exposing all whatever you did in a nice and reproducible way. So I'm pretty much done. I would like to thank the CTH for the mentoring and the sponsoring of my fellowship, of course, the colleagues at the InBio, at the Bioinformatics Division for the many inputs, the IC team, so the joke. But I love working with these guys. We've been working in remote together, and it's been such a compelling experience. I can just recommend to do it. If you want to see these packages, they are just available over there. And if you want to get a live demo of any of these, you can just type in this address, unless the port is blocked. But I think you can just try it out. And I am happy to take any questions.
All right, let's thank Federico.